Welcome to Talking Academia. Today I'm talking to Josh. Josh is a PhD student in zoology at the University of Cambridge. Hi Josh.
Hi Ola.
Thank you for coming.
No worries, thank you for having me.
So, what is your research about? What are you looking at in your PhD?
So basically I look at freshwater mussels and specifically their parasites. So there's been a whole lot of research on freshwater mussels in general because they're really important. So people don't really appreciate that, the mussels that they eat in the day-to-day life, those are all saltwater mussels. But there's also a whole lot of freshwater mussels that live in lakes and streams and rivers and things like that. And they have a whole lot of really important functions which are super underappreciated. So for example, they stop erosion, so they form these massive mussel beds along the edges of banks and stuff. And so if they weren't there, more of the bank would erode into the water with storm surges and things like that. They also help nutrient filtration, so they can take nutrients out of the water, and then because they're filter feeders, they filter it through themselves, and then they can deposit it into the sediment, so it helps invertebrates and other animals, small larvae like that, things to grow, and so then fish can feed on that, and fish can grow up, and then other bigger fish can feed on those. So they're sort of the basis of the whole ecosystem in terms of moving nutrients through the system and making sure that it's a nice healthy ecosystem. So in many ways they're a bit like the herbivores of the river world. So they take, for example, they take the grass, which is algae or other nutrients in the water, and they can make those accessible in a form which otherwise wouldn't be accessible to bigger predators. So obviously things like lions and tigers, they don't eat grass, they would eat sheep or cattle or whatever. Much in a similar way, mussels can be eaten by fish and stuff, but obviously fish can't go and get raw nutrients out of the streams. So mussels are sort of like that bridge between primary production, algae and things like that, and then the other things in the ecosystem. So they're really, really important. And also they just provide other things like protection for baby fish. So because they provide a really heterogeneous habitat, so it's not like just silt or mud, there are all these mussels poking out, so there's lots of holes and gaps and spaces for things to hide, so that way they can little fish, baby fish can grow up there without being predated and help like the recruitment of populations through time. So they're really, really important, but often underappreciated. And so they haven't been studied in terms of their more specific biology aspects very much. And so one of the things that's really missing is parasitology. So there's been evidence that a whole lot of parasites, and I'll call them endosymbionts, because we don't actually know what the effect of these things that live inside them are. So parasites is a very negative term, typically. People think of parasites and think, oh, that's really bad. So lots of the things that we see inside freshwater mussels, we don't know if they're bad or if they're good or if they have no effect. So sometimes I might say parasites, sometimes I might say endosymbionts, which is a more general term, just meaning like things which live inside something else. So all these endosymbionts, they've sort of been characterised a little bit, but not to a very large extent at all. And for most of these, we still don't know what their effect is. So basically the purpose of my PhD is to bring more attention to this really understudied aspect of these freshwater mussels, because there's evidence that these mussels, their conservation could be really harmed by these parasites. So for example, there's one parasite, or set of parasites called trematodes, which are little like worms. So have you heard of things like schistomiasis? It's a human disease. Things like roundworm and ringworm and stuff, those are all caused by trematodes. So they're very, very, very common set of organisms, and mussels are one of the intermediate hosts of trematodes. So they won't have adult trematodes inside them, they'll have some of the larval stages of trematodes. So the trematodes will use mussels as like their first host, and then maybe the larval stage will be in the mussel, and then that will leave the mussel and go to a second host, which might be a small fish, and then that will go to that small fish will get eaten by a big fish, and then the adult trematodes will be in the big fish, lay their eggs in the big fish, which then gets excreted by the fish, and they find their way back to the mussel, and the life cycle starts again. So it's a really, really complicated life cycle and really interesting, which is why one of the reasons it hasn't been understudied or is so not well appreciated, because it's just really hard to study. But what these trematodes do is they fill the gonad of the mussel, so the reproductive system basically, and they basically castrate the mussel. So they use the mussel's own resources to basically grow themselves, these trematode larvae, and they basically fill the gonad so then the mussel can't reproduce. and that's basically a lifetime infection. So once a mussel is infected with these trematode larvae, it will have those for life, and every year then, instead of producing its own babies, the mussels will just release more and more. They're called cercariae, which is the history stage which leaves the mussel and goes and swims and finds a fish. So every year from then on, the mussel will just produce those trematode life history stages using its own energy instead of its own babies. And obviously if that's a really common thing in populations, or if it becomes more common, that could cause local extinction or even species level extinction of mussels if they're not breeding enough. So that's just one of the examples of how trematodes, or parasites in general, can have a really negative effect. And those are the sort of things that I study.
How did you get interested in this field?
So I think I've always been interested generally in relationships between organisms. So I'm really interested in how one organism interacts with another organism to produce the patterns that we see in nature. So actually I started off in my master's degree, I was fortunate enough to look at coral reefs. And so for coral reefs, I was studying the symbiosis between corals and algae. They're called symbiodinium, the little algal cells which live inside corals. And so those are the things that photosynthesize and basically provide nutrients to the coral, and that's how corals can survive. So that was obviously very mutualistic. The algae get benefit from the coral because they can live in there, they can survive, they have protection, it's nice and safe in there. And obviously the coral gets a benefit because they're getting food, they're getting nutrition. So that's very, very mutualistic. And so I had the opportunity to study that. And so obviously now coming to Cambridge, I can study a more negative or maybe parasitic symbiosis. So it's still looking at the same sort of theme of relationships between organisms and how it affects the wider ecology, but it's looking from a different angle. So, but I've always been interested in those sort of relationships.
And when you work on these projects, do you collect your own data?
Yes, so for my master's, I went to Timor-Leste, which is a small island just northwest of Australia. So I was diving on the coral reef there, so I got to go down, collect small bits of coral. Then I would take them to my field station, which was basically, we set up a table and a little rough lab going there on this amazing island called Taro Island. And we would extract the DNA there, and then I would take the DNA back to New Zealand and then look at the genetics of the symbionts there. So that's what I was doing for my master's. I was studying the genetic diversity of the coral symbionts. And now that I'm over here, I do the same sort of thing. So I go out into the field, I collect mussels. So actually in January, I'm doing sampling every month at the moment to understand the temporal changes through time. So in January, when we got to the river, it was frozen over. But we're pretty tough here in biology, so we just used our net and we smashed through about a 2 centimetre layer of ice, just pushed the ice out of the way and still sampled our mussels. So it's often an adventure doing sampling in biology, but no, it's good fun.
Sounds very, very fascinating and definitely like an adventure. Your master's and your undergraduate, you did them in New Zealand, right? Yeah. So how similar are the programs if you compare them? I'm not sure if you can compare like across master's and PhD, but in general, like how similar are the programs that you've been in?
I think they're quite similar in terms of basic structure. You go to undergrad. It's a bit weird in New Zealand, so we have a 3-year undergrad, which is generally quite a discrete thing, and then honours or masters is treated as something different, as like a postgraduate addition to your undergrad, which not many people take up, and then from there you can go to a PhD. Here it seems quite different in that it's almost expected that if you want to do academia, you sort of tag an honours onto your undergrad, and then you can go straight to PhD. So the honours program in New Zealand is a bit weird. It's only recognised in New Zealand or Australia, so that's why I did a master's in New Zealand as opposed to an honours, because if I wanted to do a PhD elsewhere in the world, my honours wouldn't have been recognised, but my master's was, whereas here an honours is basically the normal stepping stone to a PhD as opposed to a master's. So in that way it's a bit different, but I think most other things it's quite structurally similar. The only other thing that I think is quite different, which I found quite unusual here, is that it's organised almost the opposite way around in terms of postgraduate organisation to New Zealand, in terms of research groups. So here it's, zoology, and zoology is broken up by the technique that you use as opposed to the organism that you study. So there's very discrete genetics groups and physiology groups and conservation groups, which is where I'm currently based. Whereas in New Zealand, it's based more on the organism that you study, rather than the technique that you use. So, for example, in my previous lab group back in New Zealand, there was people working on physiology, people working on conservation, people working on genetics, all in one lab group because we all worked on corals. But coming over here, there's people working on the same animal but in very different departments because they look at it from a different way. So that's been quite hard for me actually, having a quite molecular background and being very comfortable doing quite sophisticated lab techniques. So I arrived in my lab and there were no pipettes, not only in my lab but even in my building, just because it's very conservation based, so they're looking at a more macro scale, so they don't do much of that sort of lab work. So it's been quite unusual for me to sort of adjust my thinking to be able to talk to lots of conservationists where I currently am, which is great, but also have to really adapt to this new way of looking at things, in terms of everything has a very discrete place as in terms of the technique that we use. So that's been quite different from New Zealand. I think that's probably been the biggest change that I've noticed in terms of the structure of postgraduate programs or research in general here.
Mm-hmm. Why did you decide to come to Cambridge?
Because I'm really interested in conservation as well. So the reason I'm interested in interaction between organisms is that I love to see the patterns that they then produce. So obviously that symbiosis between corals and symbionts, that's why corals can survive in such tropical waters. Because people love diving in the tropics, right? That's because the water is really clear. So from a human point of view, that's amazing because it's beautiful, clear water. But from a biological point of view, that's really bad. Because it's really clear, it means there's nothing there. There's no nutrients, there's nothing for anybody to eat. And yet these tropical environments support these amazing coral reefs. That's only because the coral reefs have this symbiosis with this algae, which can get energy from the sun and then give that to the coral, which forms the basis of these ecosystems. So then algae can grow and fish can come in and recruit. And so all of that is driven by that symbiosis between the corals and the algae. So that's a really amazing example of how an interaction between organisms can produce this amazing global pattern that we see. And so coming here was really cool because I love to see the conservation implications of these sort of things. And there's more conservation organisations in Cambridge per capita than there is anywhere else in the world. So that was just a really natural step for me to come here, And obviously there are worse places to be than the University of Cambridge. So and there's actually a scholarship in New Zealand which provides for 3 people a year to go to Cambridge. So I thought I'd give that a go, and I was fortunate enough to be able to receive that. So that's how I'm here now.
What's the scholarship?
So it's called the Wolf-Fisher Scholarship.
Uh-huh.
So it was originally designed for New Zealand teachers to go elsewhere in the world and develop their teacher skills. But then in the early 2000s, it was also set up to send students to either Oxford or Cambridge.
So can anyone apply, or is it just zoology people?
I know it's for absolutely anybody. So it's interesting. I probably shouldn't rag on my own subject here, but there seems to be more zoologists and people like that who get it than social scientists. And that's really interesting because obviously it's an interview process, and part of the interview process is basically justifying your research and telling them why it's important and basically why they should invest in you going to Cambridge. And I think as zoologists and biologists, we're quite lucky often is that we have a very clear sort of end point, and we can very easily show the impact of our research. So for example, with the coral reefs or with the freshwater mussels, they have a massive impact on their ecosystems. If they weren't there, massive bits of nature could viably collapse, and we're seeing that already with climate change, and coral reefs are. And so that's very easy for us to explain why our research is important. And I think social scientists sometimes suffer a little bit, or classicists or people who do English literature, not because their research is any less important, but it's just because it's not as intuitive for non-experts to be able to see the effect of that. So the scholarship is for absolutely anybody, for any area of research, but it does seem to mainly go to scientists as opposed to other people, which again is lucky for me, but I think there's definite value in other subjects, and that needs to be recognised on a broader scale as well.
Does climate change play a big part of these topics that you're interested in being maybe more relevant today?
Yes, I think definitely. And again, as I was just saying, climate change has become a bit of a buzzword. So anything to do with climate change, people go, oh yes, well, we know that's happening and that's really important and it's really bad. So anything studying the effects of that or how it might influence nature automatically becomes quite important. So definitely helps.
Is this also part of why you're interested in that it's so relevant today?
Yeah, it is. So especially with the parasites, it's really interesting. So people often see parasites as purely bad. They think, oh, well, parasites are just bad and evil and we should get rid of them. But actually there's a lot of evidence that parasites can be really good. So they can actually help community coexistence specifically by harming one species but only a little bit. So often this is frequency dependent. So there might be lots of one population or a species of one population which has lots of parasites, and as that population builds up and gets bigger and bigger, the parasites can get transmitted more easily, so then they have a cumulatively bigger negative effect on that population, and so they bring it down again, and so that allows another population of a different species to be able to exist with that first population. So if parasites weren't there, it's very possible that that population would grow until it used all the resources, outcompete a second species, and so that ecosystem might only have one or two species in it. But if we've got parasites, each controlling their own species, then we can potentially have 5 or 6 species all living together because their populations are controlled by their own parasites. So that's in sort of natural, normal ecosystems, parasites can be really, really good. But with things like climate change, they're starting to disrupt that. So for example, with the trematodes, going back to what I was saying before, that larval stage which lives inside the mussels and then gets released, there's evidence that with climate change they grow a lot faster. The trematodes can produce more cercariae and they'll also live for much longer. So that means that there's more cercariae coming out of the mussels and they've got more time to find a fish before they die. So there's thousands of cercariae being released and only 3 or 4 might make it to their next fish host just because that's the way that it is. But if they can survive for even longer, we might get, rather than 1% making it to the fish host, we might get 10% making it to a fish host, which doesn't sound like much, but over a couple of generations, that's massively magnifying the parasite population, which then feeds back to the mussels, which means more are getting castrated, which means that population may die. So what may once have been just a natural sort of symbiosis with positive overall ecosystem effects, because of climate change, might turn into this really, really negative effect, which leads to extinction of mussels, which leads to extinction of insect larvae living in the stream, which leads to extinction of small fish, which leads to extinction of big fish, which leads to massive economic downturns because people can't fish anymore, things like that. So that's one of the really insidious things about climate change that people don't appreciate, and that's why these sort of ecosystems and parasites need to be studied, because right now we don't even have an understanding of how frequent these parasites are in populations, whether there's a different sort of parasite in different populations. Are they population specific? Are they very generalist? So if we moved more mussels in to try and supplement a population, could those parasites infect those mussels as well, or would they be okay? These are sort of things we don't know. And so with climate change causing all these widespread changes, these sort of issues become really important, and that's why I'm really excited to be able to be studying what I'm studying.
Mm-hmm. I know it's probably been discussed a lot nowadays, but what can, and maybe it's good to go through it again, like what can an individual do to sort of not contribute to climate change that much? How can we, like all these terrible things that you talked about, what can I do?
That's a really good question. I'd say the main thing is be aware, and if you're aware of climate change and understand that it's human driven and that it can have negative effects, I think that's the first step, because so many people don't appreciate that. And I feel like at education institutions like this, like at Cambridge or my old university, it becomes very intuitive, or it seems natural, that everybody knows about climate change and knows that it's bad and knows that we have to manage it. But actually that's not true, and there's still so many people in the world who don't believe in climate change or believe that it's happening but it's not human driven and it's just natural cycles and things like that. So I think one of the main things that we can do is to be aware that we still need to raise this as an issue. It still needs to be, talked about and addressed. And if you meet someone who doesn't believe in it, try and, convince them in the politest way possible that it is a real thing and it needs to be addressed. So one of the ways that you can do that, they love to talk about natural cycles, and say one of the classic things is saying, well, it was hotter than this 10,000 years ago, and everybody was fine, so why are we getting upset now? And the thing is, yes, that may be true, and there have been times in the history of the world where it's been much hotter than this, and there's been way more carbon dioxide in the atmosphere, and that's sort of where people stop. But what they don't appreciate is that those sort of cycles happen in the order of hundreds of thousands of years. So organisms on Earth and ecosystems have a really, really long time to adapt to those. But the spike that we've seen and we're experiencing now has happened in the last 100 years, essentially. So this is clearly not a natural cycle. Yes, it hasn't been as hot as it has been previously, but the pace at which this has happened has meant there's been absolutely no chance for adaptation by life on Earth. And that's why coral reefs are dying. That's why photosynthesis isn't happening as efficiently. That's why there's all these negative effects now. That's why the ocean is becoming a lot more acidic way more quickly than it has in the past. So in terms of understanding things like that can be a massive step in helping people understand climate change and maybe getting that critical mass necessary to convince politicians that we need a lot more than what's currently being done. And of course there is other actions that we can take in our day-to-day life. There's been an amazing study showing that eating more vegetarian meals, can help reduce your carbon footprint, because vegetables are far more sort of climate efficient in terms of their carbon footprint than eating meat or cattle is. So you can do things like that. Taking less flights. They've worked out like your carbon miles for a year and basically how long you can fly in a year to keep it to a manageable level, which is a bit of a shame for me, being from New Zealand. I actually break my carbon budget for the year on one flight back to New Zealand. So, but it's things like that you have to weigh up in terms of what sort of contribution do you want to make to the world. But little things like that, I think that those will be small steps which overall can maybe in the future have a big influence on how we deal with climate change.
It's great to have those arguments. I would definitely use them with scientific backup next time. Let's talk a little bit about not your research work, but your work here in Trinity. Or I don't know if that's considered work, but you ended up being our BA rep for the first years
yes.
at Trinity. How did this happen, and what is this?
So I did a similar role back at my old university. I was co-vice president of the Postgraduate Students Association there, and I quite enjoyed doing that because it allowed me to have an immediate impact and see the effect that I could have. So I feel like, especially in sort of science research, it can often feel like we exist in a little bubble. So we'll go and we'll do our lab work, and we'll do some genetic analysis and send it off for analysis, or we'll just do some dissections and get some raw data which is sitting there. And that while that's interesting to us, it's like you don't feel like you're having any major effect on the world at that point. You're sitting in your little bubble, and you're like, well, what is the point of this? And you know that at some point there will be some output. There will be a research article. You will be able to say, well, we need to do A, B, and C because of the patterns that I've seen. But at the time, it can often feel quite isolating. And that's why I like doing a sort of volunteering role or a representative role, because I can immediately see the effect that I can have, organising a simple event where people come and have fun and meet new people and smile and joke and laugh. That's really meaningful to me, because I can see that I'm having an immediate impact. I've made, in some tiny way, people's lives slightly more enjoyable, and I think that's really important. So that's basically how I got into it in New Zealand, and then coming over here, there was an opportunity to be the first year rep here, which is basically, being in air for first years, organising small-scale fun events, and I thought, that sounds like me, so I thought I'd give it a go.
Hey, you're doing a great job at this. Thanks for being our rep. You're also doing park run events, and you're organising board games.
Yes.
How did you come up with that?
So I was fortunate enough to sort of just stumble into parkrun, or run into parkrun as it were. So parkrun had been going for quite a while, which I think is an amazing initiative actually. So for people who don't know what parkrun is, it's basically every Saturday morning there's 5Ks organised all around the country and all around the world in fact. So I think it started to be in New Zealand quite recently, but it wasn't in Wellington that I knew of when I was there. But it's this great social event where people of all ages and abilities come together to do a 5K. And that's really cool, because I've been running ever since I was really, really young. and I love that sort of social aspect of running and that feeling of you're always competing against yourself, but doing it with lots of other people. You don't feel alone, and everybody's supporting each other, and it's just a really great sort of environment to be in. So I'm very keen to, and as I have been, organising more people to come to parkrun. So I think my friends are getting sick of me, actually. I talk about parkrun a lot, and every time I meet someone new, I'm trying to encourage them to come along, so that's good. But I've really been enjoying that. And then board games as well. It's another one of those, simple social things where people can maybe get a little bit competitive while still having a bit of fun, meet new people. And I like board games because it's very not intimidating. Some social events, if you want to meet new people, if it's just talking over drinks or a coffee, it can be sometimes a little bit awkward. You're not sure what to say. But if you've got a board game or something there, jokes and banter happen quite naturally, all in the context of the game, and then that can lead to more meaningful discussions. So that's why I want to do board games as well.
Both great events. Would recommend to anyone who's at Trinity to take part. How do you balance all that? You're doing so many things. Your research, being the rep, board games, parkrun. How do you have time for everything?
I think a little bit of planning is always good. So I try and treat my PhD like an 8-hour-a-day job, at least. So sometimes I know there'll be a bit more than that. For example, when I go and collect my mussels, they need to be processed as quickly as possible. So I'll spend 16 hours a day in the lab for 4 days, just getting all of those done. But I know what I'm going to sample, so I can block out that time and let people know. Basically, I'm going to be incommunicado, no contact at all for 4 days. But as long as people know that and I can plan my events around that, that's fine. So it's just about knowing your limits, don't overcommit, and things like board games and parkrun are very, very easy. I don't have to do too much organisation. I can basically just turn up, and whoever shows up, join with them. So yeah, not overcommitting, planning a little bit, and just knowing your limits. So I don't find it too hard to balance. And again, things like board games and parkrun, they're not chores, right? And even my PhD, that's what I feel like something really important to realise, is that there seems to be this overriding attitude at universities, less so here, but especially my old university, that if you're not having a miserable time, you're not doing it right. There seems to be a general attitude among students that, PhD sucks, work, I'm so busy, yeah. And it's like, I feel like we need to move away from that attitude, because I love my PhD. I love going to work every day and reading new things and dissecting my mussels and finding interesting things, and it's just really, really enjoyable. So, and things like parkrun and board games as well, they're not work or chores or something that I need to fit into my schedule. They're something that I choose to fit into my schedule because I like them. So in many ways, it's balancing one enjoyable thing with another enjoyable thing. So it's really not too bad.
That's a very nice positive note to end an interview on. Thank you very much for coming. Thank you for being our student rep and so positive and organizing, and good luck with your research.
Thank you so much. Thank you for having me.
Thank you for watching. That was Josh talking about his time in Cambridge, his research. Next video coming out already on Thursday. And like, subscribe, comment, share. Now we also have Facebook and Instagram, Talking Academia, so follow us everywhere. Thank you. Bye.